Well, thank you very much. It's a pleasure being here this morning, and it's always, let's say, a joy for me to share vaccine manufacturing to a large group. So I'm going to talk about managing the needs of vaccine manufacturing in Asia for improved manufacturing operations. So what I'm going to touch upon is really what I think may be helpful in terms of moving vaccine development and to the point of lights through within the Asian market, and some of the points in terms of innovation. I'll give some examples. So the outline is really talking about the environment, let's say the current environment for vaccine manufacturing, and looking for opportunities and challenges facing the vaccine industry, especially in Asia. Identifying requirements and challenges in product development through clinical stages. So I'll talk a bit about, not a lot, but a bit about each of the stages of clinical manufacture and what's needed. And then give two brief case studies on strategies for accelerating process development, really looking at balancing cost and quality and maximizing efficiency. So, evaluation of vaccination. So again, I don't want to bring this to a low level, but really vaccination is the 20th century's greatest public health triumph after improved access to safe drinking water. And more than 3 million lives are saved in the world each year with the availability of vaccines. But unfortunately, many vaccine preventable diseases still kill 3 million people a year around the world. And a lot of it is due to just the availability and access to the vaccines. What's interesting, if one looks at over the last 2 centuries on the growth and the number of vaccines that have been put on the market, one quickly sees that it's really around the technology that drives the number of vaccines that have been on the market. And since about 1980, we see a large jump in the number of vaccines. And currently we're seeing a lot of them around the immunotherapeutic approach, again generating within cancer, HIV, looking at the possibility of a number of vaccines that will enter into that market. So clearly innovation drives the development of vaccines and the ability to develop those processes for vaccines or diseases that currently have no vaccine for. It is has been, let's put it this way, has been and potentially still is a lucrative market. You can see from 1995 to 2010, it's about 5 times the market growth in terms of world sales of vaccines. So $17 billion in sales in 2010, and expecting 21 billion in 2015. And what you see with the numbers is that over the years, you're seeing a slower growth rate. And obviously, as was mentioned in terms of the number of pharma companies, especially pharma companies, first of all, buying vaccine entities, and each of the major pharma companies now has a vaccine division, and moving those, let's say, moving their, either through acquisition or through collaboration into the Asian market to access that, let's say, growth potential. But it's really an industry where there are many challenges. So we're in a business in which it's high cost, and I'll get that into in a few minutes. There's high risk in terms of developing a vaccine. There's traditionally been low investment. We see now that the investment is, there's more and more investment into vaccines, because that's a function of the value that's seen and the growth opportunities of the vaccines, but it hasn't been like that in the future. There's a few suppliers of raw materials, and overall, because of the infrastructure required for vaccine, it is a fragile network, if you will. It's a heavily regulated environment, and because of that, many static products exist, and also static processes. So it's very difficult in a highly regulated market to make significant changes in your process without having to redo some type of bridging clinical trial. And in some cases, one avoids innovation because of the time that it takes and the cost of doing that. Scientific and medical environment. As they say, all the easy targets have already been achieved. And now what we have is disease and targets in which are more difficult and require higher level technologies to be able to develop those types of vaccines. It's getting more and more of a compliance revolution in terms of the quality that's required in terms of licensing a product with the ever increasing safety expectations. And last but not least, it's a very competitive environment. Other big pharmaceutical companies such as Sanofi with Sanofi Pasteur, GSK, Merck, Pfizer, Novartis, etc., are all competing for the same piece of the pie, if you will. There's new biotech companies that are evolving, and again, the dependence on having new technologies. So just briefly about Sanofi Pasteur. Again, it's a world leader in vaccines, one of the world leaders in vaccines, which has the vaccines for over 20 diseases. It has been growing and now currently has 12 production sites, 12 production and R&D sites, so mainly from France, the US and Canada, but expanding into Argentina, China, India, Thailand, and we have 3 new facilities under construction in Mexico, France, and in China. And obviously we have an ever increasing world presence in terms of the vaccine development and manufacture. One of those I'll talk about just briefly is the acquisition of Shanta in India as a platform to the south in 2009. And the acquisition of Shanta was really driven by one, giving market access, especially to the southern hemisphere, and potentially to provide high quality, affordable vaccines to satisfy unmet medical needs, and offering adapted to UNICEF and Gavi segments, and really driven for India and middle income countries. And Shanta offers a complementary vaccine range in terms of pediatric combinations, hepatitis B, cholera, etc., and has its own R&D pipeline, such as complementary products such as the HPV and the rotavirus. So it's really a complementary acquisition in which Shanta offers Sanofi Pasteur let's say, new products in their pipeline, as well as offers a new market entry. So this slide I'll probably spend a little more time on, because I think one of the question that was asked in coming here was to really talk about what are the opportunities and challenges in operating in the Asian market. Now, when we look at opportunities, we see what are, let's say, the traditional opportunities, and a lot of which we've heard over the last day, which is low-cost manufacturing and therefore low-cost product development. You have emerging markets with large potential growth, and you have quote-unquote lower quality levels in a relative sense. But that is really outweighed by the challenges when one wants to enter into the market, and primarily by, let's say, startup companies or companies who want to do it alone in Asia. You have infrastructure and expertise which requires high maintenance, and by that is it's not a robust infrastructure that supports the whole value chain of vaccine development, and you don't have, let's say, relative access to expertise. You have higher development risks because of this, and you have regulatory challenges and higher regulatory scrutiny. And in our experience, we've seen that in some cases we found the Asian regulatory requirements and what they're asking for at even higher standards than the FDA. Manufacturing consistency is not always there. The affordability of vaccines is quite interesting in that you want to make affordable vaccines, but the affordable vaccines, which offers sales in the lower income market, really results in price erosion. And also we have the increased competition that we talked about. Putting this all together creates an interesting symbiosis between external partners that come in here and existing Asian companies, in which local manufacturers' challenges and the market attractiveness makes external partnerships or acquisition a mutual benefit for both, and as you see, is on the rise. What I'd like to talk about now is just change a bit and talk a bit about product development and give you a big picture, not to spend a lot of time, because I really want to spend a few minutes on the examples. So it takes about 8 to 12 years to develop and license a product on average, and costing close to $1 billion for each product. And the success rate, if we look at the antigens or specific targets at the preclinical stage, is less than 10%. So if you're working on 10 antigens or 10 targets at the preclinical stage, the likelihood is you'll only have one that'll reach the licensing stage. And it's going to cost you $1 billion and even more from recent reports that I've read to get that product on the market. So obviously speed and cost are the essence in a competitive environment, really, let's say, fostering the notion of efficiency and operational excellence. And again, just to break it down, the key component is really around the clinical evaluations. The size and the number of clinical trials that are expected are increasing, and with that comes a cost for greater than 50%, 60-70% of your total cost coming through clinical evaluations. And if you don't get it right at this stage, this is where you're going to spend a lot of your money and reap no benefit. Very briefly, as you go through process development, you're really looking at identifying critical unit operations. As you get into the phase 1 clinical lot production, you're talking about process definition on these critical unit operations, leading to scale-up and production of phase 2 clinical lots, and starting into process validations and starting, let's say, larger scale engineering runs, and for phase 3, doing the manufacturing consistency and eventual, hopefully, product launch. And throughout this, you're doing physical-chemical characterization to demonstrate product comparability as indications of process scalability. And one of the uniqueness, because we've heard a lot about biosimilarities at this, it's very difficult, at least with today's technology, to have a biosimilar vaccine, which is one of the, I guess, barriers to entering the market, or to, let's say, barriers to generification of any of our products. But it does make it very difficult as you enter, because anyone who enters the field has to go through these steps, even if they're making the same vaccine as a different company. So I'll go through a number of slides for each of these phases, and the intent here is to really say there is no right answer, there's no wrong answer. It's to the level that you want to do. It's really based on your company strategy, the cost, the efficiency, and the type of innovation that you want to implement. So for phase 1, the question that you will ask is, how well do you want to define the manufacturing process during phase 1? Is it good enough to get representative material to the clinic? Is it robust enough that can be easily scaled? And I think the question there is really around the antigen and the risk of that antigen being successful. And if it's really driven by a proof of concept, you probably want to take the minimum route there. If there's high confidence in that product, then you probably want to take the most robust, as I say, robust process. How much phase 1 material should you produce? You know, is it minimum needed, or do I do repeated batches to fully characterize the process? During phase 2 and phase 3, what degree of scale-up should I do at each stage? Should you utilize the phase 2 manufacturing process to support phase 3 clinical development as well? And we're seeing that as more of a common practice, where to get to the market faster, people are sort of blending phase 2 and phase 3 together in terms of doing their efficacy trial. Facilities. You know, facilities and utilities are a major constraint. But the requirements start from phase 1. So they are really no more rigorous for phase 3 than for phase 1. So you're making a very significant investment in facilities and utilities right from the start. And this has implications, as I say, on the facility investment that you want to make. Implications of equipment, instrument, systems requirements. Requirements are rigorous, again, from the start of phase 1. Implications of validation requirements. Anything to do with product safety are required for phase 1. So again, a lot of investment is going on early in the stage of the clinical development of a product. So projects are managed in the context of their environment. So industry pressures, company return on investment, project portfolio, all drive the decisions you're going to make. The quality requirements are rigorous from the start and is really driven by a need for safety. And it doesn't matter where you're marketing it, where you want to position this development, that remains the same. The quality requirements have implications regarding GMP facilities and development, the ability to handle multiple products in a facility, the ability to handle different scales of the manufacturing within a facility. And all through this, you're really looking at balancing cost and time. Get to the clinic as soon as possible, minimize time, further developing a process to minimize changes and repeating qualification, validation efforts, minimizes the cost but extends the process development. So I said I wanted to spend a few minutes on some case studies, strategies for accelerating process development. And this is by no means an exhaustive list in terms of the innovation. It's really looking at what things, let's say simple things, if you will, that can be implemented to really reduce cost and save time as you're developing a product. So again, I had mentioned that, the clinical development stage of 6 to 8 years, there's not much you can do about it. There's not much, you can change your clinical protocols, but they're getting more and more difficult in terms of what's being expected. Really, where you can save time is around the preclinical development of your process and development of processes through your clinical, which really is around, again, the point about process development in the 2 to 4 year range. The first example will be bacterial fermentation, process scale-up. So in this, we're really looking at, in the course of doing a fermentation, we typically start at a 2-liter scale to do some work. We go to a 20-liter scale, most likely to do a phase 1, sometimes a phase 2, depending on productivity, and then increasing to 200 and potentially 2,000-liter scale. And historically, what's happened is that one expects that when you operate at the 2-liter scale and develop a process, that process behaves at the 20-liter scale. But invariably, it doesn't behave the same, and you're redeveloping the process at the 20-liter scale and redeveloping it at the 200-liter scale and potentially redeveloping at the 2,000-liter scale. And we can see here that throughout this, you have not only the fermentation, but you have the concentration of your cells. You have, typically we operate with E. coli recombinant expression systems, so disruption of cells, filtration and concentration after disruptions, 0.2 sterile filtration, and then passing down to downstream for the platform for purification. And again, and then we can operate for 20 liters for early clinical trial material or non-clinical safety studies, and then 200 liter for scale-up. And again, what I had mentioned was that we see as we go from 2 liters to 200 liters, typically what we see is a significant reduction in protein or productivity. So we're seeing up to a point where we're losing 50% of cell growth or expression of the protein when the operating conditions are set to be the same. So the same process you're using at the 2-liter, you use at the 200 liter. And very simply, what we did is we developed a scale-down model, the 2-liter scale that can operate very efficiently and effectively at the 200 liter. And this is by tuning the fermentation, in which we use the same KLA or the amount of oxygen flow and agitation rate within the fermenters that gives us the same KLA value. And you can see that when we're operating at the same KLA value, either 2 or 200 liter, that we get the same product cell growth and same productivity of antigen, which really means that as we go through the scale-up process, we don't need to redevelop. We basically can go from 2 liters to 200 liters and eventually 2,000 liters in confidence in terms of the productivity of the system. Other things that we're doing, process automation, so we're implementing a lot of robotic systems, a lot of online monitoring and in-process control systems, closed disposable systems, so going away from your stainless steel bioreactors into disposable units, and a lot of isolator technology, because some of the products that we're currently dealing with are live viral products, and we have to maintain let's say, a isolator environment type throughout the whole process. The second case is around disposable systems, closed systems for purification of viral vaccines. So just to give you a quick view of how we go through our purification, obviously once through the fermenter we get the harvest coming, let's say the homogenized harvest coming from the fermenter. We do the downstream purification in terms of whether it's cell culture medial removal, looking to removal of adventitious agents, host cell protein, nucleic acids, leachable studies, endotoxin, etc., in which using a number of filtration systems, column chromatography, etc., to get the purified bulk. So you can see here that, for a specific platform approach for purification, harvest, virus release, extract purification, DNA fragmentation, purification, polishing step, concentration of buffer, and then final formulation. And obviously standardizing each of the unit operations in a fully closed disposable purification system adds to both cost and time saving in staff training, process development, equipment qualification, and process validation. So the point about if you are a startup company, this is probably the route you want to take. A lot of, let's say even Sanofi Pasteur, our existing infrastructure is based on 2,000, 10,000 liter bioreactors, and even in the early, you're tied into equipment that you've purchased, expensive equipment that requires a lot of maintenance and revalidation and requalification year after year. So basically, what we operate is closed systems, use of sterile connectors, use of tubing sealers and tubing welders, and sterilizable sensors. Disposable containers are all in bags. These bags are received gamma irradiated, and installation, maintenance, and decontamination are easy and fast. And again, this is just a schematic, but here's just a sort of what it looks like in the lab in terms of the TFF operation, permeate bags within the scale, recirculation pumps that goes, you got a retentate bag, you got a buffer bag, you got a concentrate bag, and then you have your final bulk bag that is received. And it's worked very consistent, and like I say, the time and cost that's saved by doing something like this is very significant. Fragmentation of host cell DNA by DNase, again, just setting up the whole system in terms of bags and operating with peristaltic pumps and sterilized tubing with connectors makes it a very quick, efficient system to set up and to dispose of once completed. So for closed systems, obviously functionality of all the equipment is demonstrated. So from a purification perspective, it's worked extremely well. It's proved proper design of single-use assemblies. The results are equivalent product yield and purity that we've seen, let's say, using conventional methods. Product sterility is confirmed, proof of concept for purification process and sterilized equipment. Operated as a closed system, and it really has advanced our ability to accelerate process development. And I would be remiss at the end if we don't do just one slide for advertising, which is really if our business is developing products and vaccines, and I'd be more than happy to just talk to anybody of the possibility of collaborations and partnership interests with Sanofi Pasteur. Thank you.